So good morning, everyone. I hope everyone is awake and you're ready for the tools part. So as we saw from yesterday, a lot of you are actually interested in all the different prediction tools that we have available at IEDB. So this is what we are gonna to present to you today. I will start with MHC Class 1 binding prediction tools. Sina will continue later with Class 2 tools. So this is the schedule for my talk. So I will start with a short introduction on MHC molecules, MHC Class 1 molecules, and then we'll move to MHC Class 1 peptide binding prediction web interface that we have available for IEDB. And we will also give you some guidelines on how to choose different prediction methods, how to evaluate the results, so choose different thresholds, and also different HLA alleles. And we're gonna have 2 exercises today. So a couple of you emailed us your specific questions that you're interested in using the tools. So I hope most of them we're gonna cover just by our talks and also exercises. Of course, it's not gonna be your specific sequences and your specific problem, but it should apply to your case as well. If you still have more questions, you're more than welcome to ask, and we will also have individual sessions later, as you know, so you can get more attention from us. So let's move on to the introduction. So as most of you know, MHC Class 1 molecules are expressed by nucleated cells, and they present antigens to CD8 positive T cells. The binding groove of MHC molecules is closed at both ends, which makes that the limitation for the peptide length, which can be presented by the molecule. So that usually the peptides that are presented by MHC Class 1 molecules are between 8 and 15 amino acids. In this picture, you can see that MHC Class 1 molecules have alpha chain and a small beta to microglobulin part. However, only alpha chain is the one which is determining the binding of a peptide. The interesting thing about MHC molecules is that they are highly polymorphic, and there are thousands of different variants of these molecules that exist. So finding all the possible interactions experimentally between peptides and MHC molecules is, of course, highly cost-intensive in terms of money, labor, and time. So this is why we make prediction tools for that. And prediction methods, they facilitate the selection of potential epitopes, the peptides that can actually bind to MHC molecules. From a large set of peptides, what you can do then, you can predict the ones that are most likely to bind. How we do this is basically that we have peptide binding data, experimental binding data, and Bjorn presented this yesterday as well. So we have peptide sequences, and we have binding affinities of each peptide, so we can actually use these large sets of data to train machine learning methods, such as artificial neural networks, for example. And when we get a prediction tool, which can be later on used to run predictions of unknown peptide sequences or new peptide sequences. So the data we have, binding data we have experimentally available now at IEDB is covering 126 different MHC Class 1 molecules. That's mostly human, different MHCs from humans, then we have macaques and other organisms as well. The predictors were retrained in the middle of the year 2013. However, we addressed this issue recently as well of retraining. However, the data set, new data was not, there was not a significant amount of new data. So basically, the main retraining is still considered to be in 2013. If you want to look into these data sets, different that we have available to train our tools, this is the link for that. And now I'm going to walk you through the tool itself, user interface for MHC Class 1 tool that we have. Maybe I would ask you not to go through it now at the moment just with me, so I'm going to walk you through quite quickly. So if you just look into different steps, and later you're going to have exercise where you can do it yourself and ask more detailed questions if you have any trouble. So yesterday you saw all this side. This is the main IEDB page. You saw all this side. And today we are moving on the right side. This is the analysis resources, and all the tools are here. T-cell epitope predictions tools you can find in this section, and MHC class 1 binding tool is the first one here. So if you go to the main page of MHC class 1 binding tool, you will find this is a main submission page, and this is what you're gonna use all the time if you wanna do any MHC class 1 predictions. I'm gonna come back to this one the most. I just wanna go through all of these tabs that we have up here. So if you go to help, and this is actually applicable to other tools that we have available as well. So people might not repeat this, but this is available for any tool we have. So if you go to help, you will actually find a tutorial for each tool. So this is a tutorial for MHC class 1 binding prediction tool. And the tutorial basically explains, and this is what I'm gonna do it as well, explains step by step on how to actually make a submission and run the predictions. So you can see there are different steps, and down there, if you scroll down, you will see that each step is explained in detail. You also have an example tab here, which basically just gives you a chance to quickly check if a method is working fine. So you just choose one example, one of these, press submit, and this data will be inserted into the tool, and the predictions would run. Under the references, of course, you will find the list of papers that describe different tools. So if you want to find more details about each and separate methods, you can read the publications. And also this would be a list of references that you have to cite if you used the tool for your studies. We also have software available that you can download to your computer. So if you want to really run large calculations and large predictions, we suggest that you download software packages for that. So each tool can be obtained from this webpage, and this is free for academic users, and there are licenses available for industry as well. And of course, we have a contact page, so if you run into any trouble, you can contact us, and we will be happy to help. So this is just getting back to the main submission page, and I'm just gonna walk you step by step on how to do predictions. So if you come to this page, basically this is the place where you put your protein sequence in. So your protein that you're looking for epitopes within that, and you can put it in different formats. So it can be a space-separated sequences. It can be a one continuous sequence, or it can be a sequence probably which is most common in a FASTA format. So then you can also choose a file from your computer and upload several more sequences if you want to. And you can always, of course, put in more than just one sequence as well. And basically the important part here is that the format of your sequence is usually detected automatically by the tool. So if it has this sign at the beginning, it will be treated as a FASTA file or FASTA sequence, and otherwise it will be treated as a continuous sequence. So that just says that you have to be careful. If in case you put the sequences separated by spaces, you really have to specify that. Otherwise, spaces will be ignored. But I suppose that FASTA format is the most common that everybody uses now. And then when we move down, you can see there are prediction methods available. And this is the list of methods that we have available now. This is like 7-8 methods that we have now online. And IEDB recommended is one, as the name says it, we recommend it to use. So if you are always questioning which one do I choose and what's the best, just use IEDB recommended, because we are choosing them for you. We are checking what's your MHC alleles you're interested in, and then we are choosing the best available method for that MHC allele. So in this case, when IEDB recommended is chosen, consensus method is gonna be used if MHC molecule was part of a training set. So it means if we had experimental data available for that molecule, and NetMHC pan is going to be used, which is basically one of these pan methods that Bjorn mentioned yesterday as well, that are capable to extrapolate to new MHC molecules as well, even if these molecules were not part of a training set, if there is non-training data available. But I'm going to come back to the methods a bit later and give you more detail as well. So then you choose also species that you're interested in, and then finally you can choose your MHC alleles here. So there is a list that you can actually, automatically it's marked that the list is showing only the most common alleles that we have, but you can always untick this box and you will see all the possible MHC molecules that are known. And then you're also choosing, of course, peptide length that you're interested in. The most common for MHC class 1 would be 9 and 10 MERS. You can also upload, if you want to run the study for like really long list of alleles and the multiple lengths for each allele, then you can upload a file yourself prepared in this format, where you have an allele name and after the comma you have a peptide length that you're interested in. The results that you see afterwards.
Question?
Yes?
Sorry. This upload allele file section, is this where you can upload some of your reference tables for representative populations?
Yes, so you can put whatever list of alleles you want to.
I know there are some reference lists.
Okay, no. So we have a reference list down here, the one that we recommend, you mean? Yeah. Yes. This is a reference set of alleles. Yeah, I'm gonna present that a bit later, but yes, this is in this section. So if you click this, it will give you a suggested list of reference alleles that we have ready for you. Yes. The results can be sorted, and if you wish, and it can be sorted, for example, by percentile rank, which I'm going to present it as well, or you can just leave it the results as that the peptides are in the sequence as you uploaded them in your protein sequence. And also, if you want to do some filtering, you can filter based on predicted values, and you can have two different formats of the output file as well. So once you hit submit, you will be seeing the results page. Basically on top, what you have is your sequence or sequences. If you uploaded several of them, each sequence gets a number or a unique ID, which later you can see in the table as well. So you can actually see where the result came from, which sequence. And in the table, what we have is you have allele, you have start and end position of a peptide, peptide length, and peptide sequence itself. You will also see which method was used, and also you will get a percentile rank. That's the score that we're using at IEDB, and it's a uniform score among all different methods that we have, and this basically means that percentile rank score for a peptide is a percentage of randomly sampled peptides scoring better than the peptide in question. So we are taking a large list of random natural peptides, like 100,000 of random natural peptides. We include your peptide into that list, and when we get predicted values, predicted affinities for that, sort based on the best affinity and see where actually your peptide ranks. Okay, so the smaller the score, the smaller the percentile rank score, the better binding the peptide is. Yes?
Is there a way to enter a virus, a whole genome, rather than a number of individual
protein sequences? No, at this moment, no. You have to enter all protein sequences. I mean, you can enter multiple protein sequences, okay. Yes, but it has to be protein sequences separated by IDs. Yes, if you go and click on expand the results, there is a small tick here. Especially if IEDB recommended method was used, it tells you which actually, it will give you the results of each individual method as well. So if you can see here, the consensus method was used, and the consensus method, as the name tells, consists of several different methods. And in this case, it's ANN, SMM, and combinatorial library method. And it gives also separate results for each of the method if you're interested in, because you know the details of each method. In terms of using just one specific method of your choice, you would get actual IC50 values predicted as well, if you wish to. So how do you actually choose from all this large list of possible methods that we have? So if you look into all the prediction tools, we can actually divide them into two different categories. And as mentioned before already, there are allele-specific methods and pan-specific methods. So these allele-specific methods are the ones that are giving predictions only for that MHC molecules that were part of a training set, so that we have experimental data for. And in terms of pan-specific methods, we go beyond that, so we're extrapolating to the new MHC molecules as well. And this is basically in the nature of different methods, if it's possible to do it this way or not. So for allele-specific methods, we have these methods available. We have SMM, two different SMM methods. We have COMLIB, and then there is one artificial neural network-based method also available only for allele-specific predictions. And there is a consensus method falls into that category as well. And in terms of pan-specific methods, here we have mostly neural network-based methods, because these methods are capable to extrapolate to the new molecules. So we have NetMHC pan, we have Pickpocket, and NetMHC cons. The motivation for consensus method was basically based on the thing that seeing different predictions of different methods, combining them can actually give higher performance than any of the individual predictors. And studies were done on that, and when we noticed, and we decided to try and combine different methods that we have into consensus method as well. So in our case, consensus method at IEDB webpage combines NetMHC, SMM, and COMLIB. And of course, in this case, if you're combining different prediction methods into one, you want to have a uniform score. And this is where we use a percentile rank score. And as I mentioned before, the percentile rank score you can get for any method you want. So how the consensus method works in this case, so if we have, you can see we have the stable results from before. We had the consensus method used on all these separate methods that were actually combined. So if you take one, you will see ANN get one score, SMM got another score, and the third method got the next score. So how do we actually choose which one is used as a consensus final score? In this case, we take the median of the three methods. So as simple as that. And the IEDB recommended method, so consensus method will give predictions only for the molecules that were a part of a training set, okay? And then, as I mentioned, we have these pan-specific methods. So the IEDB recommended method, what it does, it actually uses consensus if a molecule was part of a training set, and it uses NetMHC pan if it was not. And this is based on the benchmarks that were done before, basically just seeing how the performance of these methods are. And of course, recommendations may change when the new benchmarks are done and if we see different performances and different ranking of the tools. Any questions to the methods? Yes. Can you use the mic, please?
Do we have a method for predicting epitopes for the non-classical MHC molecules like QA?
Yes.
Is there any method that we can use?
No, here we don't have. You cannot upload. Yeah, that's a very common question. You cannot just, yeah. And you cannot upload for now, I believe. But that's been a request just to upload the sequence, MHC sequence, protein sequence. For itself, it's not available at the moment.
Thank you.
Yes.
For a lot of molecules that you have a range of peptide length, how do you compare the rankings from 8, 9, 10, 11-mers to each other?
Maybe you, Bjorn, can answer that, right? You just did the length study.
I would think that's it's actually an open research question. So far, it's just based on the IC50 values, and so essentially the predicted IC50 values are used to rank across the different lengths. But we think that's at least if you want to predict T-cell epitopes, probably wrong, because while the longer peptides actually do bind with reasonably high affinity, they tend to not get made. So we are working on a correction factor for that. Talk to Thomas. Where's Thomas? There. Okay.
All right, so this was on how to choose different methods, and now it's going to be more on actually how to choose different thresholds on what to consider a binding peptide and what not. So there are several different ways, and also it depends on you usually on what's your study problem, what are you looking for, how many epitopes are you interested in, how many peptides you can actually check or use in many cases. So for MHC class 1 molecule, in terms of IC50 values, it has been, several studies have shown before that actually 500 nanomolar is the threshold to define binders. And the ones with lower affinity than 50 would be considered high binders, and the ones with lower than 500 would be intermediate binders. And then if you go more, then of course the binding is reduced. So this ensures, if you use a binding affinity as a threshold, this ensures you that you have a reasonable affinity, at least intermediate affinity, when you consider your peptides as binders. Another way to pick your potential binders or potential epitopes would be to choose top 1% of the best predicted peptides for each MHC and length combination. So that would cover most of the immune responses, and this case you would ensure that you would get equal number of peptides per allele. And also the third thing would be, which is not exactly the same as just taking top 1%, that it might seem, but would be to use a percentile rank score as a threshold. So take, and in this case we suggest you take peptides with a lower percentile rank score than 1. So there are several different things to consider, and it always also depends on your problem on actually how many peptides you want to choose, or you could choose. It also has been checked, and this is done by Sinu in a study a few years ago, basically that we discovered that there was different alleles might have different number of binders. So it's not the same for different alleles as well. And you can see it in here that for if we have all peptides, the percentage of binders would be different in different MHC class 1 molecules. And also peptides with a higher predicted binders, alleles with a higher predicted number of binders actually have epitopes with stronger affinity as well. And so this suggests that we can actually have allele-specific affinity cutoffs as well. And this was done before, and what we have is that we have a list available. So this is, if you get back to a help page and check the tutorial, you will have a list there which shows allele-specific thresholds that are suggested based on the studies that have been done. And our recommendations on choosing thresholds would be actually any approach is good, so it really depends on you, on what are you looking at. So use affinity and ranking, both are reasonable approaches. It also would make sense to combine the two. So you can take, for example, top 1% of peptides, but with a lower affinity than 500 nanomolar. Then you are really sure. And of course, you can always just look into allele-specific thresholds as well. And now on how do you actually choose a representative allele. So this, of course, if you have a specific study where you're having specific donors and you're using their specific set of HLA alleles, that's probably already clear for you. But in some cases, it's difficult if you're maybe working more on a global vaccine, actually on how do we choose MHC molecules. Because as you know, if you only look into humans, there are all these different MHC molecules. So there are like more than 6,000 different HLAs available in humans. And as I showed before, we had data, experimental binding data for about 80 of them. So that's just 1.3%. So this is really not a large amount. And then if you look into more, then there are different regions in the world have different frequencies of HLA molecules as well. So different populations will have different MHCs that are more common in that population. And you can find these frequencies in the allele frequency database if you would like to. So all these problems would actually were solved by making a reference set of alleles, which was a question already about that. So we actually have 27 alleles that are recommended because we are the ones that cover 97% of population. And this would be the list of recommended alleles. So if you are looking into development of a global vaccine, this would be the alleles you want to cover in your study. And in this case, if you get back to a submission page, you will see that here you can click on using the allele list, and you will get all this list imported for you automatically. So this is very easy. Yes?
A question for you about that 97% figure. Does that mean that 97% of the population has all alleles from that list? Or
No, using all of, if you would have all of these alleles would, is it the same? It would cover 97% of population.
so 97% of the population would have one of those?
Yes.
Yes.
I see. Thank you.
Okay, so that was it from what I had to actually say, and we are perfect on time because we have 2 exercises to do in half an hour. Do you have any more questions before we move on to exercises? Yes?
Is there a reference or link to information about the method for deriving that list? Because I assume that even though it covers 97% of the population, some are better covered than others with that list.
Yeah, is there a paper? There should be, right? In list, there should be somewhere.
So the data comes from allelefrequencies.net, and we did the calculation at some point. I think it was in the last dengue epitope mapping paper where we did it the last update. But this is, there's nothing very fancy about it. It pretty much comes directly from these frequencies.
Okay, yeah, but I think there is a link in IEDB to the paper somewhere. I can try help you find later. Yeah.
Thanks.
Okay, so this is the first exercise, and I hope we can do it in less than 10 minutes because the second one is longer. So you all get to try. I hope you all have your computers. So basically we have an AMA1 protein from Plasmodium falciparum, and this has been tested as a good candidate as a multi-antigen malaria vaccine. And there is this paper down here, if you want to read it later or have a look at it now. But basically, there were volunteers immunized with a vaccine containing the protein, peptides from this protein, and the ELISpot responses were tested later. So we know one 15-mer, which was tested in donors, and then we know the set of donors' alleles. So what you have to do is, within this 15-mer, find the best epitope which would be binding to this donor. Okay? So you have to use MHC class 1 prediction tools. Try to do it yourself now in 10 minutes. I will be around trying to help, and Sino can help as well. And we're going to go through it in 10 minutes. Let's go through this exercise and see if you all get the same. So there is apparently a bug or a problem with getting Excel table exported, but it's good in this case you don't actually need this because it's a small table. So I just want to go through this with you. If I try. Okay. This one. Okay. So basically what you had to do in terms of your protein sequence, you only have a 15-mer peptide, right? So it's not a large sequence. We only want to test this peptide for potential 15-mer for potential epitopes within it. So if we go to the tool, our protein sequence, we just paste this long peptide here. Okay, so for the methods, you don't actually even need to think anything. You just use IEDB recommended. We have humans as a species. And now for how to choose alleles. So basically you can choose alleles one by one. And for each allele you also choose a peptide length. So in this case, okay, we have O201. So you can just go down the list, you see O201. And then you're capable of choosing different peptide lengths. But in this case we don't know, right, how long the best epitope could be. So we can actually use all lengths and get predictions for all of them. And when we do the same for other alleles, 2601, So it adds alleles like in a list. If you chose a wrong one or you don't want to include it anymore, if you press this button here, it will delete it from your list. Very simple. 1801, B1801. And I believe it was 44. Can you still hear me? Okay. 4402. All lamps. Okay. And it's, of course, now we want to check and get the best epitope, so we actually sort the results on the best percentile rank score. If we submit, and hope this doesn't crash now. Now everyone should stop checking web Facebook so I can get the results here. So I believe normally there shouldn't be any error with Excel file, right? Yes, and we did try this before and it should work. Yes, there is a question?
So usually in the case of vaccine development, you are not using a peptide to search against those 27 alleles, right? You are using the whole protein.
Yes.
So how do you come up with a
oh no, so here I just chose one peptide sequence from this paper, but if you go in the paper, you will see that we actually test all possible 15 mers overlapping by 11 amino acids.
Isn't it too difficult to come up with one single epitope of that?
Oh, this is just an example. I mean, this you can do for all the protein sequence if you want to. This is just an example I chose for you to have a short sequence.
So how do they come up with a decision to make which epitope to use?
They experimentally tested all peptides overlapping the protein sequence. This was one that came up positive. So they wanted to know what the minimal epitope was within the 50 mer that was recognized in order to make tetramers. So this is actually a real example.
Okay, thank you.
Okay, so we cannot get the results here. And of course, that always happens when you try to show something live. But it's good I have a solution here for that. So this is what I already did, and the results should look like this. And I believe most or all of you got these results. And if you look, in this case, it's already ranked by the best percentile rank score, so the best epitope would be the one on the top of your list. And in this case, it's a 9 mer, and if you look down, you can also see that it's binding even more than to one allele very strongly. So that would be your candidate, and this was actually, if you get back to the paper, you will see that they tested when experimentally and confirmed that there were a T-cell response to this epitope. So yeah, the ones who got the same result, you can be very happy and proud about yourself now. Okay, so this is the next exercise. I hope nothing crashes because this one is a bit more complicated. So imagine that you have to design and this is just an example, okay? This is not how you would normally do this, but you have to design a dengue vaccine that would work for across the world, so a general dengue vaccine. So in this case, I just give you a sequence, so use this strain from the gene bank, so you can find the protein sequence. And you will have, normally you will have probably to use the 27 allele reference list that I showed you, right? That covers the most of the population. But because, as you can see, things don't work out perfectly when all of us try to do the same. So in this case, please just use one allele, this one, okay? That's the allele from the list. And only look into peptide lengths from 9 to 10. And you have to answer the following questions based on your results. So how many peptides would you choose if you follow top 1% rule? So choosing top 1% of epitopes. And how many peptides you would choose if you actually looked into percentile rank score. And at the end, and this is a very good example as was repeated to you yesterday, there are already known epitopes in IEDB database. So check how many actually of the top 10 peptides are already known to be epitopes in IEDB database. So you can use something that you learned yesterday as well. And we have 10 to 15 minutes for this exercise. Okay, So everybody managed to get the results for this one, more or less? Who finished? All the numbers? Yeah, you did, Bjorn? Good job. So who has all the numbers and answers to all questions? Okay, cool. At least someone. Okay, so I will try again. It will probably will not work out. Go through this in live version. So we get back to the same submission page. I cleared it out. Yes, okay, so we have to find, first of all, gene bank is not part of IEDB, so you actually have to go to their web page. The best is to Google. Google knows everything. So if you paste the gene bank ID here, you will get the sequence. So the protein sequence is down here. Sometimes it depends, I guess, on the browser also. If you copy paste just this sequence, it might give you trouble. So the easiest way is actually to find the FASTA file for this and copy paste from here. So we take all protein sequence and go back here. We paste it in here. Don't pay attention to all the spaces. This will be ignored by the method. We leave IEDB recommended in this case, and now we have to choose this one allele and choose peptide length 9 and the same allele again and peptide length 10. We want to rank it on a percentile rank score because we will be mostly looking at the top best peptides. So now fingers crossed. Stop running. Oh yeah. So in this case, a lot of you ask why you didn't get this nice pretty table as before. So usually if you have a really large set of results at the end, you will get the text file. So the table is not available anymore. In this case, the best, and you don't get the option to download as Excel, but I believe if you just copy all the data, you should be able to paste it to Excel table. So just have to do paste special, I think. Didn't work. And yeah, choose text. So now you get, each column separately. So these are all your results, and if you can see if it's ranked based on a best percentile rank score here for each peptide. So the first question was asking you, I keep opening wrong things. What would you choose, or how many peptides would you choose if you were following top 1% rule? So in this case, basically what you have to do, you have to know how many peptides in total you have, right? And just choose top 1%. So if you do some tricks here in Excel, you can see the count is here, and that's 6,767 peptides. If you take 1%, you will take 67 or 68 peptides based on this number, right? Who got the same number? Yay! 20% of people. And then the next question, but you understand why, right? The ones who didn't get, do you understand now why this number? Okay, cool. Now actually the different thing would be to check for the percentile rank score threshold. So using 1 as a threshold, and we get back to our results here. So in this case, it's already, as you can see, it's already ranked on a percentile rank score, so we just have to go up to 1, and this will be all, and that's, if you can see here, I don't know, it's a very small number, it's 24, right? So it's this less. So in case you really can only select very few epitopes, in this case would be maybe a better choice for you. And then the last question was, and I don't know how many actually of you managed to check these, but we have to go back to IEDB and check the top 10 peptides, if they are actually known as dengue epitopes already. So if I take top 10, let's see, and I can just copy paste sequences from here, go back to IEDB main page, we are here again on a curation side. We are looking for a linear epitope. We paste the sequence, and we see if something is already available in IEDB. In this case, it's not. Let's take another one so that we find a hit and we are happy. So if you go here now, paste another sequence, update your search, and this one it is known. Yay! So how many out of 10 did you find, the ones who managed to do it? 5. Yes, that's what I did as well. 5 out of 10, top, best. Yes, so basically that would be it. And now you can choose the best epitopes, design a dengue vaccine, and be a very successful scientist. So thank you. And if you have more questions, I have a few minutes now, and then we have individual sessions later on as well. Any questions? Okay, thank you.